So the recording is in progress and we are ready to go. So then let's start with an introduction. Hello, everyone. Thank you so much for tuning in today. It is a hot day, so I'm very happy that you still made your way to our lecture series. And we are super excited to have Jeremy today with us. And it's a real pleasure and like a real honor that you are speaking now at our lecture series. And it's really interesting because maybe a year and a half ago or so, I was working on a bigger algorithm on trying to incorporate different cell mechanics into a machine learning framework, which didn't really work out. But at the time, I tried to find as many resources as possible, like what other people have worked on cell learning before. Then obviously you can't miss Jeremy's work. So since then, I've been, well, more or less harassing him to join our lecture group. So I'm very happy that you've agreed and are now sharing your work with us. To give you a quick introduction about Jeremy's work himself, like whenever you Google him and you look up his CV, he mentions that he is a mathematician by trade, but by pure coincidence found his way to biology. And his background is really remarkable. So like he did his PhD in algebraic topology, like nothing to do with biology at all, at the Trinity College here in Cambridge in 1981, excuse me, please. And then for transition time, he worked at the Ali Dixon Institute as an instructor at the University of Chicago. And at the same time, at least is my understanding, he worked as a research fellow as well in pure mathematics at the Trinity College as well here in Cambridge. And at that point, he started to become more and more interested in complexity and complex systems when he started to volunteer in teaching computer science. And this kind of apparently marks his start into diverging into complex systems and analyzing also biological systems as well. Then he had a quick change to the industry. He joined HP Labs at Bristol in 1987, where he also became the director of basic research in Europe in 1994. But he continued to remain linked to academia. So he had academic appointments basically throughout the US and Europe. He had one in Stanford, MIT, and Cambridge. He was a professeur invité at the ENS in Paris in 2000. And he also became an appointed member to the UK's Engineering and Physical Science Research Council between 1999 and 2001. And then in 2001, he left HP to become a visiting scientist at the Bauer Center for Genomics Research at Harvard. And this is roundabouts the time when they completed the sequencing the human genome, and I presume that also initiated creating loads of genomics research and institutes around the world. And in 2003, he joined then the newly formed Department of Systems Biology at the Harvard Medical School. And for then pursuing his new ideas for cellular learning, he moved last year to Barcelona to the Universitat Pompeu Fabra as a distinguished professor. And there he has his own lab about cell learning, which I presume he's talking about today, at least based on the title, right? And obviously, it is unnecessary to mention that he has endless numbers of publications in very high class and highly ranked journals, with some of them over 200 citations. So it's really, really impressive to follow your background and to follow your work. And as I said already in the beginning, we are super happy to have you today coming to our lecture series to present your work. And with no further ado, thanks very much, Jeremy, for being with us today, and the floor is all yours.
Thank you so much, Leo, for that introduction, and Gautam for organizing this very interesting series of talks and for inviting me. I'm delighted to be here. I mean, I was listening to you tell people about my history, and I suppose the thing that occurs to me is how random this must seem, and certainly from my own perspective, it's been a series of random accidents. However, they've been extremely fruitful and interesting. So I'll tell you about the sort of latest turn in this stochastic trajectory, which has to do with the question of cell learning. And I hope everyone can see my slides on the screen here. So I thought what would be helpful, since I don't know, I think people come from a lot of different backgrounds in the audience. So I thought I might start with one of the starting points for me in thinking about cell learning, which is the work of somebody whose name I think has faded out of the historical consciousness, Herbert Spencer Jennings. He was a American biologist who did a lot of very important work. He was extremely well known in the earlier part of the 20th century. And in particular, around the turn of the 20th, about 1900, he summarized much of his work in a book on the behavior of the lower organisms. And this is an amazing book. And he describes many remarkable experiments, but one experiment in particular was on a unicellular eukaryote, one of the clade of ciliates. This particular one is known as Stentor roselli. Stentor comes from the Greek for a trumpet shape, and it's a single cell that typically anchors in marine in aquatic environments to an algal holdfast, and you can see it represented in the picture. I don't know. Can you see my cursor on the screen? Yeah.
We do.
Okay, good. It's named for the little hairs, cilia that project from its cortex, which rotate, and this creates a vortex in the surrounding fluid that brings bacterial particles to what is in effect an oral cavity, and that's its mode of existence. And what Jennings described was a remarkable hierarchy of avoidance behaviors that Stentor exhibit to repeated irritation. And the repeated irritation was that Jennings would squirt it with carmine dye particles through a pipette. And what Jennings describes is that when the organism first encounters this, it tends to bend away, as you can see again on that picture. And if you keep repeating it, it might bend more. But then it changes tack, and it exhibits a ciliary alteration, which you can exhibited there by the cilia around the sort of rim standing on end there. And the effect of that is the rotation is altered. And so the vortex, instead of bringing particles to the mouth, ejects particles. Okay. So if you keep irritating it, it might do that a bit, and then it then changes its behavior all over again, and it does an extremely rapid contraction down to its holdfast. So the ciliates are a very interesting clade. They're remarkable behaviors that they exhibit, but in particular, their cortex is kind of part muscle, part neuron. They exhibit an action potential, and this is part of this very fast contraction. And if you leave it be, it will kind of then sort of re-emerge. And if you keep irritating, it might contract again and might contract again. And then, and I have to say, it's very hard not to use anthropomorphic language when you describe this. It basically gets really fed up. It pulls up its holdfast and swims off. Okay. Now, keep in mind, this is a single cell. So I heard about this first, actually, in a lecture of Dennis Bray, who's shown in the bottom here, when I was just making the transition from computer science into biology. And I'd been, I knew a little bit of biology at the time. And Dennis was giving a talk, and he flashed a slide up about this. And I was just completely flabbergasted, because I'd never thought that individual single cells could be capable of some behavior like this. And I talked to Dennis about it afterwards. He wrote a very interesting book, which I think anticipates a lot of the things that we are now thinking about and trying to do, called wetware. And he talks about the ciliates there. And this behavior has ramified, it's sort of resonated over the years, and many people have sort of picked up on it. And one of them was Oliver Sacks, who wrote about it in a New York Review of Books article on the mental life of plants and worms, among others. And Sacks extricates a quotation from Jennings, which I put in the middle of the slide here, about Jennings's view about this kind of behavior, suggesting that the behavior even of these single cells was of a level of complexity that could be compared with that of animals. Now, there's a long kind of history associated with this. The person who's much better known from that period of time is not Jennings, but his adversary in thinking about the behavior of cells, German biologist Otto Lieb, and he took a much more what one would call reductionist stance on the behavior. He believed that essentially all animal behavior, even the behavior of single cells, was just simple chemical reflexes, if you like. And Lieb is often seen as being the sort of forerunner of behaviorist psychology and is remembered for that. And Jennings's ideas have been largely forgotten. Anyway, I was immensely struck by this, and I started digging around and trying to read Jennings's papers. And then I discovered, to my horror, that actually Jennings's results were considered not to be reproducible. And there was, in fact, a community of people, they were rather small, particularly from the 70s and 80s, who had started thinking about aneural organisms in the context of neurobiology. And I started talking to some of them, and one of them, David Wood, who's a very good experimentalist who did a lot of work on the ciliates, in an email said to me, well, it's all very nice what Jennings says, but it can't be reproduced. And he referred me to this article in the Psychological Record, which was not obtainable electronically. And I was really disappointed. I thought, but something about this, I eventually went and looked for the paper because I couldn't square this with reading Jennings, who's a really good scientist, and his papers are very lovely. And I had to go into the library and photocopy it, which was a strange experience because I hadn't done that for years. And I have to tell you, and you can, I'll send you a copy of the paper if you want, but this is the single worst paper I have ever read in my life. Okay, and it is bad on so many dimensions that we would waste a lot of time even discussing it. But one dimension is that they couldn't find stentor roselli, which actually is not a trivial point because it's quite difficult to find. And so instead they used a different stentor, a cousin of stentor called stentor ceruleus. And stentor ceruleus is very similar, but unlike roselli, which is typically fairly sessile, ceruleus is typically rather motile. And what they found, which is what they say, is that when they tried doing this, it kind of swam off. No kidding. So on the basis of this completely ridiculous attempt at reproduction, the scientists in the 70s and 80s basically considered Jennings's work to be not reproducible. Okay, well, I was definitely not happy with this, and I think I made a real nuisance of myself in group meetings, trying to persuade people in the lab that we really needed to go back and do Jennings's experiments again. And, I have to say this went down like a lead balloon because most of the people in the lab want to do molecular biology, not stare down the microscope and do kind of 19th century natural history. Or they wanted to make mathematical models of cells or whatever. And then eventually, over time, the right people came to the lab, right in the sense that they were crazy enough to actually think that this was an interesting thing to do. One of them was my postdoc, Sudhakaran Prabhakaran, pictured now a little bit older than he was at the time. He's now the co-founder and CEO of a startup. And my PhD student, Joseph Dexter, who has a wide variety of interests, including now the digital humanities, and has been in Macau and is now coming to the Queen's University in Belfast. So it was the two of them working, basically in their spare time over a long period of time. And I won't bore you with all the technical problems we had in putting together an experimental setup where we could duplicate the experiments. And this is a video. I hope you're seeing the video. This is stentor anchored to algae. The little shake that you saw there was a pulse of not carmine, but the particles we use from the pipette that's just on the right. You can see the bending away, and you can probably see, I hope, that kind of straightening of the cilia that is ciliary alteration. I'm really a cackhand at video editing, so I'm sorry. This is okay. So you can see now that it sort of goes back to a sort of normal shape. And then there's a gap in the video, which you can't see. You can't see the next pulse. But then you'll notice another ciliary alteration. And then bang, there's this very sudden contraction onto its holdfast. And it's amazingly fast. And then I'm afraid there's another kind of glitch in the video. And you can see that the stentor is kind of extending itself. And it's basically trying to pull away from its holdfast there. And then it swims off. Okay, and this is just one example from a whole series of videos we did. That's the table in here. This is a sort of distillation of, kind of 2 years worth of experiments. And one of the things that we found, which Jennings really doesn't talk about at all, is an amazing amount of variation from experiment to experiment. And I think when Jennings was talking about his experiments, he was kind of distilling a kind of typical behavior of what he had seen over countless years of observation. But of course, in our scientific context, we have to kind of deal with the variation. So one of the problems was, how do you extricate from all of this a statistical test of what Jennings was claiming? It's all in the paper. You can do the tests. The p-values are amazing. And Jennings was correct. There is a hierarchy. There's a lot of variation, but there is this hierarchy of behavior. Okay, so Jennings was right after all. And we did not use the word learning in the paper. I thought it was a form of learning, and in the draft that we sent to Current Biology, that word was used. The reviewers jumped all over us. I didn't know enough about learning to really fight them off, so we just retreated. And so, we call it complex behavior. But I would say it's learning. And when I found out more about this, I think one of the elementary ways in which learning is described in the psychology literature is it's a change of behavior in response to a repeated stimulus. And on that basis, I think this is an example of learning in the single cell. So that was the starting point for me of realizing that in trying to get to grips with this kind of behavior, we really needed to start thinking about learning. I have to say that it's worth pausing for a minute and just asking ourselves why it was that the scientific community basically rejected Jennings's ideas in the way that they did, because basically they shouldn't have done that. Anyone who had actually read the paper in the Psychological Record, including David Wood, who's a very good experimentalist, should have realized that it was an absolutely horrible paper. And they shouldn't have concluded from that Jennings's many papers were wrong. And I think my conclusion from the literature is really that this was an ideological problem. People really didn't want to believe in the 70s and 80s that single cells could do complicated things. And Jennings's papers just didn't fit that story, that narrative. And I'm afraid that the subject of learning seems prone to this kind of problem, because it's not the only instance of it. And I've just flashed up some papers here, but this is a reminder of a completely different episode, which goes back to a scientist called James McConnell, who claimed that simple invertebrate organism, a planarian flatworm, could not only learn, that's uncontroversial, they can learn, they can be conditioned, but also that conditioning could be transferred to other organisms through cannibalism and through regeneration. And that created a lot of interest, and it was ultimately discredited, and McConnell himself was discredited in a much the same way as with Jennings's work. And now in the modern era, people are coming back to these experiments and finding, well, actually, McConnell wasn't quite so wrong as everyone thought. So I think there are a lot of lessons in this for, how science works and how we need to also think about the kind of how we need to do very compelling science in order to counteract some of these problems. So for me, that's been a very important learning process. But coming back to actual learning, in order to sort of get into this, I had to sort of basically turn to colleagues in cognitive science. And for me, this has been a really important sort of direction to take, to realize that, thinking about this from the context of molecular biology, cell biology, signal transduction, all the things we had been doing was really not sufficient, that we really needed to draw on ideas that come from outside sort of that sort of core biology. And in particular, and I think I probably mentioned Sam's name earlier when I was talking to Leo, my colleague Sam Gershman at Harvard has been one of the really key people for me in helping guide me through some of this morass. And two other people, the neuroscientist David Glantzman and the psychologist Randy Gallistel, have been much involved in the discussions that we've had together. And this has resulted in a couple of perspective papers, which provide some background for some of the things I'm going to talk about, and particularly the paper in the Proceedings of the IEEE, which I wrote a couple of years ago, sort of summarizes some of the ideas that I'm going to tell you about today. So Leo mentioned that I've sort of moved to Barcelona, and one of the reasons for that is that this is an opportunity to really pursue this direction of cell learning. And I'm doing that with colleagues, one of whom, Jordi Garcia-Ojaro, is with us in the audience. I don't know why Jordi's heard this talk so many times. He must be thoroughly bored. But also two other colleagues, Aneta Koseska and Dietmar Schurke, who are in Bonn. And this is through an ERC Synergy grant, which has really provided the means to attack this problem in a serious way. Okay, so what does it mean to turn to cognitive science? How does that help us get to grips with this issue of learning? Well, obviously, learning has been studied in psychology and the cognitive sciences for some time. And in particular, the behaviorist psychologists in the 20th century formulated a number of protocols for studying learning. I'm sure you're familiar with Pavlovian conditioning. That's the one that everyone knows, Pavlov's dogs listening to a bell just before they get their food, and after that, the bell triggers salivatory responses. But there are simpler forms of learning. There's non-associative learning, which happens when a single stimulus is repeatedly presented, and the learning behaviors that have been uncovered for that fall into two types. One is habituation, which is pretty much what you think it is, which is, you get the stimulus again and again, and your response to it becomes lower and lower, as shown in the graph on the left there. And there's sensitization, which seems like the sort of converse to it, that you get a repetitive stimulus and become more and more response to it. And it's easy to think that these are sort of converse to each other, but I think that maybe is a bit misleading, that sensitization is a different kind of thing to the converse of habituation. But that is something we could leave for the moment to discuss later. Once you get to multiple stimuli, then you run into, classical conditioning, Pavlovian conditioning, but also something that is very much in the news there, what is called in the psychology literature operant conditioning, but in computer science is reinforcement learning. If you do a YouTube search for Skinner's pigeons, so Skinner was the person who really introduced this mode of learning into behaviorism, he has some amazing work on training of animals using operant conditioning. And if you put Skinner's pigeons into YouTube, you'll see videos of Skinner's pigeons playing ping pong. And it's a vivid illustration of how effective reinforcement learning, not just for machines, but also for pigeons. Behaviorism gives us a set of protocols for studying learning. And so one starting point for thinking about learning in the context of cells is to take advantage of this. And there's already quite a lot of literature around this. Let me focus particularly on habituation, which is one that we've studied at some length. Habituation is a lot more complex than a simple declining response. In particular, experiments that have been done over decades in animals, and by animals I mean both vertebrates, rodents, humans, chimps, whatever, and invertebrates, reveal that habituation has some kind of universal properties called hallmarks. And these were codified. There was a famous meeting of vertebrate and invertebrate psychologists and neurobiologists from both the vertebrate and invertebrate communities, which don't normally talk to each other. And they codified a set of 10 hallmarks that seem to characterize habituation across the animal kingdom. And they're listed there. And these take the form of what happens during habituation if you increase the frequency of the stimulus. Well, what happens is that the habituation response becomes stronger. It happens more quickly or is more pronounced. What happens if we increase the intensity? That frequency sensitivity is hallmark 4 here. If you increase the intensity, what happens is actually the habituation becomes slower, it's less response. And then there are other things that you can do. You can habituate, you can wait a period of time so that the habituation response declines, and then you can rehabituate. And what you find is that the rehabituation is typically quicker. So there's potentiation of habituation. So you can read these, and it's not necessary to remember all of them. But the point is that habituation seems to exhibit these very interesting hallmarks. And if you talk to a psychologist or an ethologist who's studying habituation in animals, you would have to show that these hallmarks were obeyed if you wanted to convince them that what you were observing was a true form of habituation. And it's worth pausing for a minute and just thinking of how remarkable this is, because this is across all animals. And it's across all sorts of modalities. It can be, all sorts of different stimuli, all sorts of different neural systems that are involved in this. It could be acoustic, it could be optical, and yet they seem to, for the most part, exhibit these same behaviors. So if that wasn't surprising enough, we've known for many years that actually exactly the same thing happens in individual cells. And this is a remarkable set of experiments that were conducted in the 1990s by Dan Koshland and his students. And Koshland may not be a household name to you, but if you talk to any biochemist and say Dan Koshland, they will say the Dan Koshland. I mean, he was a very famous biochemist. He did many things. And in particular, he was one of the pioneers of studying bacterial chemotaxis. And this is in an earlier stage in his life. And you can see from the review paper that I've just listed there in the bottom that he was already thinking about the behavior of bacteria and their chemotactic behavior from the point of view of neurobiology and neuroscience. And if you read his writings, it was clear that he was paying a lot of attention to what was going on in neuroscience. And in the 1990s, he started taking this and thinking about it in the context of individual cells. So again, Koshland is being very prescient here. I think he would be rather startled to see that we've taken so long to sort of catch up with him. But what he shows is that in a particular kind of cell, which are PC12 cells, these are a neuroendocrine tumor cell line. And in response to various stimuli in the actual context, they come from the adrenal medulla, and in response to stimulation from an upstream neuron, they secrete a number of hormones into the systemic circulation. So in particular, noradrenaline or dopamine. And it's the secretory response that Koshland is studying. And what he shows, and this is an outtake from his, sorry, I beg your pardon, paper, that they exhibit not only the declining response to repetitive stimulation, that is the first thing you look for, but also over a series of papers that we kind of tabulated from one of our papers here, many of the critical hallmarks, including frequency sensitivity, which in many ways is the most critical one. And we see the same hallmarks in the single cell. So if you know the universality of habituation across animals is amazing enough, it's even more amazing when you find that these same hallmarks are taking place for habituation in the individual cell, where there ain't no neurons, it's all molecules that are doing it. So there are many remarkable things about this. One of the remarkable things is that nobody paid any attention to Koshland's work. There are essentially zero citations to this in the 10 or 15 years after Koshland did this work. I don't think the cell biologists of the time, the molecular biologists, really had a clue what to do with this. And I think it's only more recently that, some of us are coming back to looking at this and seeing Koshland as a sort of pioneer in thinking about behavior learning in the context of the individual cell. Now, Koshland was a great scientist. He was a great biochemist. And one of the things he did, he contributed hugely to an understanding of biochemical mechanisms. The idea of induced fit is an idea from Koshland. The idea of allostery is also one of the things that he contributed to. So one of the things that's always puzzled me is why he didn't get into the mechanism, the molecular mechanisms underlying this. Because, in the cellular context, you can really hope to pull this apart. He didn't. And I wish, I could have had the opportunity to ask him why. Maybe he tried and didn't get anywhere. Anyway, this was something that we wanted to sort of think about. We didn't have the ability really to look at this experimentally at the time. We're very much hoping to do that now as part of our ERC Synergy grant. But we wanted to look at it at least from a mathematical point of view. We want to ask the question, well, okay, we have this very interesting behavior with all these hallmarks. Can we find any kind of molecular circuit or something, which is plausibly a molecular circuit that could exhibit all the hallmarks? And the answer is yes. And, we found several. I'm showing you one of them here. And this was an attempt to try to find something which had some biochemical plausibility. And so it's based on the idea of post-translational modification of proteins, if you like, phosphorylation, which is one of the things which is extremely prevalent in cells, eukaryotic cells in particular, bacterial cells as well. But it's a way of sort of changing the state of a protein by adding a chemical moiety to one of the amino acids, and to do so reversibly through catalytic modification. So this is I've always been fascinated by this, because there's potentially so much state available through this mechanism that you wonder, what are cells doing with this? And I don't think we've even got to really grips with this problem yet, despite all that we know. So this circuit is built out of sort of modification, demodification motifs, where a protein, a molecule moves between an inactive and an active state through a process of modification and demodification. So you've got these modification, demodification loops, and they're arranged in a particular kind of way. In this case, the active state encourages the active state of this. But in this loop, the active state encourages the deactivation of this. So it has a different sign. So what we have here is what's called in the biochemical literature an incoherent feed-forward loop. An IFF. It's incoherent because while this is positive and that's positive, this activation is a negative one. So it's incoherent. And incoherent feed-forward loops are known to have nice properties of adaptation. And in fact, they exhibit habituation. It's not difficult to show. But we could never find a parametric regime in which a single incoherent feed-forward loop exhibited frequency sensitivity. And following an old idea in the literature, which goes back to a psychologist by the name of John Stadden, who suggested that one way to solve this problem was to do a concatenation. We looked at the concatenation of two incoherent feed-forward loops, and that does in fact exhibit most of the main characteristics of habituation. There are a couple that don't fit into this mathematical framework very easily, so we weren't able to test them. And I'm showing there some of the actual plots from the paper for frequency sensitivity in particular. And all of this was done by a numerical study. So one of the things that I think I would extricate from this work, which is, I think, very relevant to the next step, which is, can we actually see this going on inside a cell biochemically, is that the properties of learning processes like frequency sensitivity are actually informative about mechanism. Because if you want something that is going to be frequency sensitive, it's no good looking for a single incoherent feed-forward loop. You really have to look for something which has some kind of concatenation behavior like that. That's, what we think. So I think that's a sign of how looking into these learning mechanisms in detail in the way that these hallmarks have been done is actually very important for sort of moving forward with this. Okay, so that's fine, and that's a kind of nice, kind of story in its own right. But there's a problem with this, and the problem really goes back to behaviorism. And you'll probably know that the behaviorist sort of orthodoxy was that you weren't allowed to sort of think about what goes on in between stimulus and response, because all of that was so quote unquote mental, and by definition, therefore, in the early 20th century, unscientific. And behaviorism was, very, very powerful. We still use behaviorist protocols to study learning. But in the end, particularly when computers came on the scene, it became clear that, well, actually, computers can do things that appear to be, very similar to how humans do things like reasoning. The behaviorism gave way to cognitive science. And in cognitive science, there's an explicit interest in what happens between stimulus and response. And so the cognitive way of thinking, I think, is a much more powerful and appropriate way for us to think about learning. So the whole issue of learning is sort of very complicated in many ways of thinking about it. But I think there's a way of extracting a definition of learning, which I think is particularly useful for us in the context of the single cell. And it's represented there in these three statements, which I think are all very plausible. An entity, it doesn't have to be a living entity, is learning if it's acquiring information from its environment. It's encoding that in what I like to call an internal representation, which is kind of like a model of the world. And then it has the possibility of using that to causally guide its behavior. So one way to think about it is that this internal representation is a kind of has two kind of faces to it. One is it's an encoding of information. It's a prediction of what the organism will do, and so for me, what constitutes an internal representation is a molecular kind of system in the cell, which, if we had access to all its states, would tell us what information had given rise to that and what the organism will do as a result of doing that. Okay, so that's the kind of test that we got our hands on a bona fide internal representation. And I think part of what we're trying to do in this synergy project that we're embarked upon is in trying to get our hands on internal representations. Leo, I should ask, how much time do you normally allow for in these talks, and how are we doing on time?
We are around 45 minutes in.
Okay.
I would say take as long as you need. We can extend the discussion time at the end. There's no problem.
Okay. So it's always a little bit sort of unnerving when one talks into a computer without any feedback. So I do encourage people, if they want to ask questions, to just jump in at any time. But I don't know what the kind of etiquette is in these talks, but I'd be very happy if anyone wanted to do that. Okay, so I'll take it a little bit further in trying to say more about internal representation. So this idea is really trying to sort of exploit the understanding of learning that comes to us from psychology, cognitive science, and bring it to bear on the cell. But in fact, I think we already know that cells are learning from this point of view. We already actually know several molecular internal representations just from our understanding of biology, the current literature. It's just that people don't think about it in terms of learning. So let me take you through a few of those because I think they help to sharpen our understanding and maybe sort of get to those kind of boundaries where people start to disagree about what's learning and what isn't learning, which is part of how we learn ourselves in how to think about this. So I'm going to tell you 3 examples, which, if you know any biology at all, you will recognize immediately because they're so well known. Every biologist knows these examples. There's nothing, kind of novel about it. It's just what's novel is thinking about them from the point of view of learning. Okay. So the first example, and it's probably my favorite example, actually, because it comes from a bacterium. And a single bacterium exhibits exactly the kind of learning that's outlined in the definition I gave on the previous slide, and exhibits this through CRISPR. And you may be familiar with CRISPR as a kind of, the great biotechnology. It's won Nobel Prizes for the people who worked on it. This is not the biotechnology. It's the biology from which we've, kind of vandalized the biotechnology. The biology is an adaptive immune system in individual bacteria. And it's really astonishing. The way it works is that an individual bacterium may be attacked by a virus that attacks a bacterium as a bacteriophage. And evolution has given certain bacteria the ability to detect the DNA of the virus, to snip out a segment of that DNA and insert that segment into the bacterium's own DNA. Okay. And it's inserted in a position called the CRISPR locus. This is actually how CRISPR was discovered, because people, when they started being able to sequence bacteria, started finding these sequences and the sequences around them, and realizing that they were bits of phage genome. And having encoded the information from the environment in their own DNA, then if the bacterium is subsequently attacked by the same phage, It can transcribe the DNA in the CRISPR locus into RNA, and then it has this lovely mechanism of RNA kind of guided DNA detection and destruction, which is really what the CRISPR biotechnology has kind of um, fixated on. Because it's such a useful way of sort of inserting things into DNA, and it's biotechnologically extremely useful. But this satisfies all the conditions that I laid out, right? It's information is acquired by the organism. It's encoded. We can see the encoding. It's actually in the genome, and then it's used subsequently. One thing to be aware of is that the bacterium is unlikely to be attacked by a phage twice in its lifetime. So actually, this system evolved not to benefit the individual bacterium, but to benefit the progeny of the bacteria, the clade. So the CRISPR locus in a bacterium is actually an encoding of that bacterium's ancestral history. And now we can actually sequence bacterial samples. In this case, in the bottom of this slide, it's an extract from a paper which did this from patient samples in a hospital, and you can sequence them, and you can see that the individual bacteria have different CRISPR loci. The CRISPR arrays look different because each of these bacteria had a different individual history, and this individual history can now be read out. So this is, I think, actually really amazing in terms of thinking about the way in which an individual bacterium has been able to record the experience of its lineage in terms of the phages, that is, or plasmids, which are transferable pieces of DNA that bacteria can exchange with each other. That is experienced, the environment that is experienced over many generations. So I think this is a very interesting example to sort of think about on many levels, partly because it's so sophisticated. And this is in a bacterium. And you begin to wonder, if bacteria can do that, what are eukaryotic cells doing? Evolution doesn't forget. There isn't an exact equivalent of this. Maybe the picoRNA system in mammalian cells might become an alternative, but nobody's really looked at this. The molecular biologists have pulled it to pieces, but they haven't looked at it from this learning perspective, which I think really can be quite powerful in helping us to sort of think about the implications of this. Okay, so that's one example. Here's another example, one that everyone knows. It's the circadian clock. Each of the cells in our body, for the most part, runs a kind of clock. There's a central pacemaker in the hypothalamus that resets to the ambient light. Evolution sort of found circadian clocks, built circadian clocks in different organisms over the history of life. They're not all the same, but this is another example of a system which encodes information. You can see that because every time you encounter jet lag, because you move to an environment where the outside time is different from your inside time, but eventually your system resets. It's learned what the outside time is. So the details of the clock are known. It's extremely complicated, transcription, translation, feedback, negative feedback loops. I asked one of my colleagues at Harvard who works on the clock, Chuck White's, how do I tell the time in a clock? In other words, what's the internal representation? What molecular states of the clock do I have to look at in order to tell what the external time is? And it's strange to me that the circadian clock biologists haven't actually figured this out. And Chuck's guess was that there's a particular protein called PER, which has a very elaborate process of aggregation in the cytosol and the nucleus, which is shown there in these beautiful cryo-electron tomographs, which he's studied over a long period of time. And his view, because this builds up over the circadian cycle, and his view was that if you could see the distribution of these aggregates between the cytosol and the nucleus, you could kind of back out what the time was. Although I don't think anyone knows how to do that from actual data. So, but I think that's probably a very good estimate of what will be happening. So here's another example of an internal representation. And cells not only require encode time, they also encode space. But it's not external space, it's internal space. It's internal space inside the body. It's the position along the body axes. And this is an amazing set of genes known as the Hox genes. And in a very nice paper, Howard Chang at Stanford, who studied these at some point, points out he uses language which is sort of very much resonant with what I'm trying to say here. In fact, he calls it an internal representation. And he points out that, so the Hox genes, there are 39 Hox genes in humans. The vertebrate lineage went to 2 genome duplications. There's been losses and all the rest of it, but anyway, there are 39. They're arranged in sort of 4 groups, A, B, C, D. And if you basically, if you take a sort of digital view and ask which Hox genes are on and which Hox genes are off, then they separately encode both the position of a cell along the anterior-posterior axis of the body, that's the C genes here, whose encoding, whether they're on or off, represents that axis, and also the distal proximal position in the limb. And that's a different set of the A genes that encode that. So there's an implication here, and that is that if I had the ability to look at the expression, whether on or off, of all 39 of these genes in a cell in the body, I may be able to back out where in the body, according to these coordinate axes, that cell lies. Now, the biologists certainly haven't figured out this encoding. I mean, they kind of talk about it in sort of metaphorical terms. Howard Chang is making that suggestion here, but nobody's actually written down the coding table. But I think this is another internal representation. Okay. So the point of all this is just to say, current biology has many examples of learning and internal representations. If we're going to take this idea forward, I think we have to do it in a much more systematic way, and that's one of the things we're hoping to do as part of this ERC Synergy grant that we're working on. And I think if I had to put my finger on what is different here to what people have been doing in all the cell biology and molecular biology that discovered all these things that I've just talked about, it's that we have to get our heads out from inside the cell, because people's attention has really been focused on the molecules. Most molecular biology is dedicated to putting very little information outside the cell, because what they're interested in is just looking inside. And that's fine if you want to track molecules, but it's hopeless if you want to track representations, because you have to put a lot of information in a controlled way in the environment, in multiple dimensions and with potentially multiple contingencies between them. And then you have a chance of asking, well, what might be getting correlated with that inside the cell? And so the situation we kind of imagine is that we have highly high-dimensional input space for the environment, and potentially a very high-dimensions output space of behavior that the cell might exhibit. Maybe secreted proteins, it could be morphology, it could be movement, it could be proteins inside the cell. And what we hope is that we can somehow get our hands on the internal representation as being a sort of low-dimensional encoding that sits between these two high-dimensional spaces. compressing the one and predicting the other. And if I put it that way, I'm sure those of you in computer science will know that this sits within the context of various forms of representation learning that have been developed in AI and machine learning, and which I think will rely upon to go down this dimension. And uh, uh, this, I think, offers us an experimental pathway for systematically uncovering internal representations that we can actually undertake both experimentally, theoretically, and computationally. And that's the reason I think that this is a good time to try. Okay, I should stop, but let me leave you with one, what's the point of doing all this, in some way? Where does it really get us? And I'm sure you can imagine dimensions that could come out of this in terms clinically and for medical purposes and all the rest of it. But I think there's something which I always have found very, very interesting to think about, which goes back to the early cognitive scientist Jakob von Uexküll, who in the early part of the 20th century coined the idea of the Umwelt, which in German just means environment. But von Uexküll invested this with a very particular meaning, which is that it's the subjective experience of an organism to the world outside. We think that we all live in a world that exists independently of us, but we actually construct a perception of it, each individually. And animals, of course, construct their own perception of it. And we all have a kind of self-centered perspective of that. That's our individuality and autonomy give rise to. And this was what von Uexküll was trying to get his hands on. And there's a famous passage that's often repeated when people talk about it, about the Umwelt of a tick, as it is going about the business of living. And I think, for me, one of the potential consequences of taking this, cognitive stance into biology is that it offers us a sort of scientifically well-grounded way to start thinking about the subjective experience that organisms have of the world around them, cells have, because internal representations, to me, are the encoding of that subjective experience. And if we can really get our hands on them and understand the kind of computations that cells are performing on them, then perhaps we can begin to kind of decenter ourselves from this, kind of perspective we have and put ourselves in the kind of shoes or mind of other organisms. And, who knows where that will lead. Okay. Thank you very much for your attention. I'm sorry for sort of prattling on for so long.
Thank you so much for your talk, Jeremy. That was fascinating and a very inspiring talk. So now I would say we start a Q&A section. So people in the audience, you're welcome to unmute yourself, ask your questions whenever you want. Please note that we record the session, so if you're feeling a bit shy, you can also text into the chat box, and I will read them out loud. So maybe for the time being, to wait whether people have questions or for them to make up their mind, I can start with a question. So I'm really interested in your definition of learning, cognition, and then also like the internal representation of a system that we don't have access to, like that acts as a kind of black box, right? So there's an internal representation. We want to test now whether this system that acts as a black box can learn, right? And what I'm interested in is, where do you draw the line between, for example, a finite state machine, which doesn't have like an adaptation per se to its environment, but rather like on an organism level, what would be the difference between a couple of pre-coded steps versus learning and adaptation to the environment? Does it need access to previous knowledge, or does it need to be newly acquired knowledge? What is your take on this?
Yeah, so I think this is a question we persistently come back to, Leo, and I think I don't think that we have a clear-cut answer to it, because I think there's always, I mean, we're dealing with living systems, so any attempt to define something is always going to run into the problem of gray boundaries, ambiguous boundaries. And that's true even if you think about, defining life. Biologists get very upset when people ask them to define life, because it ends up in a conversation where, oh, but I have a counterexample to your definition. Yeah. So I think one way to maybe sharpen the problem is, you quoted the example of finite state machine, but maybe another way to say it is that you could imagine that an entity is receiving some input, it's doing some kind of processing on that, and then it does some output. Is it better to think of that as an input-output process or as learning? How would you distinguish between an input-output process and learning? And I don't know exactly. I can offer some suggestions for how I've been thinking about it. And I think it has to do in part with the internal representation, because there are two things. One is that you would probably want this internal representation to persist for longer than the timescale of the information in the environment, right? If it's still there, then, it's not really sort of representing things. But also, it may be that you want this internal representation to persist beyond the kind of immediate output that it gives rise to. And that you might be able to detect its presence through some other form of interrogation. And this very distinction between this sort of input-output perspective and the idea that you need to have a separate way to interrogate the internal representation mirrors a debate that has taken place in cognitive science, which is, broadly speaking, between the neuroscientists, in the modern era, between the neuroscientists who basically bought into the behaviorist idea that stimulus response protocols are an adequate way to think about behavior, and the cognitive science perspective, which focuses much more on the internal representation and feels that without additional evidence that the internal representation is kind of present beyond the stimulus response that is observed, it is unreasonable or not sufficiently, it's not adequate to consider that as a form of learning, right? And one of the cognitive scientists, I think, who's particularly pushed that idea is Alan Wagner, who, if you know about the Rescorla-Wagner model, is one of the sort of pioneers of thinking about, behavior from a mathematical standpoint, but more generally from this cognitive standpoint. So my kind of reading of that literature is that the kind of behaviorist input-output perspective is okay for some people, particularly those in neuroscience who typically use those protocols. But in the cognitive community, there are those who would want to see that this internal representation has a life of its own, which is not so coupled to the stimulus and the response, in order to ascribe that as a form of learning. Okay, so is that an appropriate way to think about it in the context of the cell? Frankly, I don't know. I'm keeping an open mind. I think we have to sort of get our hands on some of these things and play with them before we will be able to sharpen this distinction in an adequate way. And I'm fully expecting that we will get to situations where, I think it's learning and someone else doesn't. And I think that's just life. That's just the way biology is. I think that's fine. I don't have a problem with that. What I have a problem with that is definitions that we can't test.
Yeah, that absolutely makes sense.
I don't want to get into semantics. If you don't agree with the definition, that's fine. Go and make your own definition. But, we should have definitions.
Oh, absolutely. Absolutely. That makes sense. I have so many follow-up questions to this, but like I see that there are people in the audience that have questions themselves. So I think, Ageda, it was you first who raised their hand. Please unmute yourself. Go ahead.
Yeah, thank you. I was thinking about how to define this internal representation when you were explaining the topic. And following Leo's question, I think that a difference between a closed system where we know the stimulus, black box, and output is variability. Like at the end, we can know that something is learning or that the input has been or it has an internal representation because there's variability in the output. So it's still a very behaviorist definition, but I think that makes more sense when we are trying to study learning in non-neural organisms, than an internal representation because it's again a definition that could be anything.
Okay. I mean, I think the idea here is very similar to the kind of informal definition of learning, which is that it's a change in behavior in response to the same stimulus. And so the internal representation is capturing that idea that in the absence of the prior history of stimulation, the behavior would be different. And in that sense, there is a variation that you're speaking of. So I think that's kind of encapsulated, at least in the way that I try to think about it. So I don't think the internal representation can be that arbitrary because, as I said on one of the slides, my way of thinking about it is that if one is going to claim that something is an internal representation, then I think we are bound to be able to say, okay, if I can see that internal representation, which means if I have access to all the molecular states, then I should be able to infer from that what environment gave rise to it, what was the information in the environment, at least in some statistical sense. We might not be able to decode the whole of all the information. The cell might not need to encode all of that information, it's just what's important for the cell. And also, we should be able to predict what the cell will do as a response to that. So I don't think internal representations can be just anything. They have to be tied to the environment that was seen and the behaviors that followed. I hope that provides some answer.
It does.
And I see there is a question in the chat box, but because Oliver raised their hand before, I think. Please, Oliver, go ahead. No, it's not the case. Okay, then Kishan writes a follow-up to Leah's question on this black box. Can we ever figure out how to uncover this internal representation? Perhaps having more measurement or getting more data and testing more models. So this is a question by Kishan. Apologies if I mispronounced your name.
I'm sorry, Leo. I kind of lost track of the question. It's how do we get our hands on the internal representation? Was that the thrust of the question?
This is my understanding. Yeah, whether we need more measurements or getting more data or testing different models, how can we access this internal representation?
Yeah. Okay. So I think I kind of mentioned on one of my slides this notion of representation learning. So I think at least one approach to this problem that we're trying to take is that we have in principle the experimental ability to subject cells to complex environments. We can controllably create sort of environments with multiple signals. We can create contingencies between the signals, so we can put probability distributions into the environment. And we can subject cells to that. And then we can look at what the cells do. And in a very broad sense, we can look at different kinds of proteins in the cell through various forms of microscopy. We can look at how they behave in the sense of how they move, whether their morphology changes, what their internal structure changes, whether they secrete things into the surroundings, as Koechlin did in studying his PC12 cells. So I think we can see input and output, and these are potentially very high dimensional spaces. So I think there are methods from machine learning and artificial intelligence which allow us, in principle, to find low dimensional spaces that provide a compression of the input and predict the output. And the techniques are, autoencoders, variational autoencoders are one method. The informational bottleneck is another process that's been also quite widely used in biology. But it doesn't really matter. I mean, there are techniques for finding these. Now, these are abstract representations. They come in the form of vector spaces with probability distributions on them. So that's only one. That's only a starting point. We have to kind of take those and sort of figure out how the molecular machinery in the cell is kind of representing that. And that's another problem altogether, for which I think it's going to be a kind of cycle of guesswork, interventions that we can make biologically, genetic, chemical, more kind of computation and a cycle of doing that to see if we can sort of get our hands on the actual molecular pieces. So I think there are strategies that we can adopt for trying to identify these internal representations, experimental, computational, and theoretical strategies we can adopt.
Yeah, really interesting. And now, Oliver, finally, it's your time. Please go ahead.
No worries. Not at all. Thank you very much for a really, really wonderful talk. I really like this idea of internal representations, but looking through your examples, you have, I suppose, different ways of presenting this. So with the CRISPR example, you are actually sort of taking a piece of information and sort of writing it down, essentially by putting it in the DNA. With the circadian rhythm, you've got the synchronization of an oscillator to a periodically changing external force, and that's sort of encoded in some ways in protein complexes. But it's I don't know the words to say it is to me. It strikes me as being a little bit different than if you're writing it down as DNA. And for Hox genes, I don't know very much about them, so I assume it's a kind of response of differentiation in response to a chemical gradient. So this internal representation, particularly with the DNA or the CRISPR example, I can see this internal representation is it's written down and then it's kind of expressed with the circadian rhythm. It's a periodically changing set of concentrations of every component that's involved in the oscillating system. So are you making distinctions here between different types of representation? How do you think about that?
Yeah, a very good question. I think we've sort of been spoiled by DNA. It's such a beautiful way of encoding information, and we know a huge amount about it. And CRISPR is a wonderful example, but it kind of brings us back into the territory that we know so well, which is why we know so much about it, which is DNA. Unfortunately, or maybe that's the wrong thing to say, maybe fortunately, I suspect that the internal representations that we will be searching for are most unlikely to be encoded in polynucleotides. Now, I mean, it's not because, I mean, I wouldn't rule out that there might be still some amazing thing going on with picoRNAs and things like that. But as far as we can tell, that's not happening. So I think the internal representations are going to be much more elusive. They're going to require this more like the kinds of things that we see with the circadian clock. they're going to be distributed over multiple molecules. These molecules might have themselves be in very different states. They may be in states of modification. They may be in states of aggregation. They may be localized in different parts of the cell. So the very nature of the representation, I think, is going to be very foreign from our experiences with DNA. And not only the substrate, but also the nature of the encoding, how you go from knowing the substrate to knowing what the information is. The nature of the coding table will be profoundly different. So my suspicion is that, we are going to have to sort of think very differently about how information might be encoded. It's going to be a much more dynamic thing. This is biochemistry. It's dynamically changing. It's catalytically changing. It's going to be probably very stochastic because we're down to sort of, we're talking about finite numbers of molecules. It's going to be spatial. I think we get a sort of hint as to what might be involved when we think about examples like the circadian clock. But I wouldn't be at all surprised if, if we manage to get down this road, we will find that actually the information is also encoded in things like, the architecture of the cell itself, maybe the morphology of the cell. There are examples, for instance, that if you look very closely at the structure of the mitochondrial network, the way the different mitochondrial bits are connected to each other, there's actually information over there about the stress conditions that the cell has experienced in the past. I would read that as encoding of information in the structure of the mitochondrial network. So I think it's very open, but I don't think we can. I think CRISPR is a very special case, and I think we're going to have to come to terms with the fact that these internal representations that we're after are encoded in profoundly different ways.
Okay, thank you. I mean, as an experimental chemist, I like that answer because it's a bit more malleable for me to actually do something with in the lab. I mean, kind of just on that though, the amount of sort of learning that you do, if it is to say the different concentrations of different proteins or different molecules that kind of within the cell, then the sort of the memory of what you've learned is going to be or sort of maximum about 5 times the half-life of whatever the protein is, assuming it's or expressed as it degrades over time. So what is the length of memory for learning? I'm not sure if I formulated that very well, but
yeah. No, it's a great question. This question of timescale, I think, is really an important one. And I think it's useful when thinking about that is to come back to our own personal experience of learning, because I think we're all aware of the fact that, in terms of our own learning, we have different timescales. We remember, something that we heard a few minutes ago, and it's still in our head. And then we remember something that we encountered a couple of days ago, and we recall it. And then there are things that we remember from a lifetime, right? And in the cognitive and neuroscience literature, we understand that these different timescales of memory are all kind of implemented by kind of different neural systems, and that information is moved between them. And there's a very elaborate process by which this information is kind of replayed in kind of forming, consolidating these kind of long-term memories. So I think it is very plausible that for the individual cell, it confronts essentially the similar sort of general problem that the organism confronts. It too will likely exhibit timescales of learning. There are things that it needs to remember for short timescales and things that it needs to remember for much longer time. CRISPR is an example because it passes that on to its progeny. And I think we can make a good guess, I think, that for sort of eukaryotic cells, the long-term memory is likely to be, the sort of epigenetic mechanisms that lock down chromatin states in eukaryotes, because we know that those epigenetic mechanisms, which are not recoding the DNA, but rather reprogramming how that DNA is expressed, that those mechanisms can be inherited by daughter cells, and passed on. So rather like CRISPR, they have the ability to, you see this in the developing organism as a cell begins to, if you like, learn what cell type it must become. It goes through these progressive stages of becoming more and more like that kind of cell and passing that information on to its daughter so that the daughter cell becomes more like that. So I would say that the long time scale is probably going to be, sort of related to that kind of epigenetic behavior. I'm very fond of post-translational modification, and I think that's probably a very good mechanism for some kind of short-term memory, because it's a lot of state and it turns over very nicely. I think there may be an intermediate timescale, and that I think gets much fuzzier as to what the timescales of that would be. I think it might depend a lot on what cell types one is looking at. I don't have a very good feeling for them. Yeah.
Thank you very much.
Yeah, I think, Jeremy, you left us all with a lot of food for thought. That is very, very interesting. I think we got now to the end of our talk. There are no more questions in the chat room, and there are no other raised hands. So with that, I will then conclude the session. Jeremy, thank you so much for joining us today. That was a real pleasure having you. And please keep us posted about how your work on cellular learning goes, how your new lab goes in Barcelona. And yeah, thank you very much again for presenting your work today. Maybe a last item. We are currently organizing our next term, so all the following lectures that are coming then in autumn. So to everyone in the audience, if you happen to know someone or if you would like to present your own work, please contact us either via the Google group, or you can just email us directly. In any case, enjoy the hot days wherever you are. Europe is quite hot at the moment. Enjoy the weekend that is coming now, and we are looking forward to see you very soon. Jeremy, again, thank you very much.
Yes, thank you.
Thank you. Thanks, everyone. And I look forward to joining you on other occasions. Thank you very much.
Fantastic. Bye-bye.
Bye-bye.